introduction. And in the next 20 or maybe 25 minutes, I'm going to give you an update on this project. It's using Chen genetic approach to control aflatoxins in corn using a novel strategy called host-induced gene silencing. Some of you may have questions, so I'm just going to address. So what is gene silencing? When I discuss the gene silencing, you will see that the small RNAs are involved. So I will introduce a little bit about small RNA, and at the end, I'm trying to address what is host-induced gene silencing, and why should we care, or what is the importance of this mechanism. Gene silencing is a natural plant defense mechanism that is present in plants. And it occurs at post-transcriptional stage. It means after the mRNA is synthesized, during the process stages, that's when the silencing happens. So sometimes we also call it as post-transcriptional gene silencing. And this process is triggered by double-stranded RNA. And double-stranded RNA is naturally occurs in the plants during certain conditions, such as when the virus infected, or when there are some pathogens, or some of the plant messenger RNAs during the processing stage, they fold back and form double-stranded RNAs. And once they're triggered by this process, they can induce a sequence-specific breaking down of the target gene. When the target gene is breaking down, they won't be able to make proteins, so they are not expressed. That's called gene silencing. And this is a diagram showing in the plant, when the genomic DNA has some certain areas, they can fold back and make a double-stranded DNA. That will trigger some enzyme called dicer. And these are some other scenarios when the double-stranded RNA are present. When the dicer is an RNA enzyme that can break them down into a smaller molecules. This is called a small RNA. It's about 21 to 28 nucleotides. And based on the oranges, they have different oranges. Sometimes they call it small interfering RNA, and sometimes they call it micro RNAs. So in the next slides, I will address what's the difference. Once they break down into this, the small bottom strand, called a guided strand, can bind to the specific target sequence. And then this enzyme called the RNA-induced silencing complex can initiate the breaking down of the target sequence, and then the target mRNA gets degraded. So the difference between the small RNA, there are two kinds. One is called small interfering RNA, the other is micro RNA. Both have similar size, about 21 to 28 nucleotides. And usually the micro RNAs are usually from the endogenous, and they are involved mostly in the growth and developmental regulations. And the small interfering RNAs are usually involved in plant defense against the pathogens. So I'm going to see the next slides. So the importance of gene silencing, this has been a very powerful tool. It has been used to study the functions of unknown genes by silencing them, see what kind of phenotype you get once you get silenced. It has also been used to study the genes in the pathogen. And one of the interesting, since this discovery, it has been used to control the nematodes, insects, and the parasitic plants. And one of the major advances of this strategy is they can achieve a specific silencing effect by just feeding the synthetic small interfering RNA molecules to the cultural media. And this has been achieved in C. elegans and also in Aspergillus nigrans. What that means the small molecule, small RNAs can penetrate the cell membrane, get into the cells inside, which we never understood how this can happen, but actually it happens. So the demonstration of the movement of small RNA between the parasite and the host plant, and between the insect and the host plant, is indicating the importance of small RNAs in the host-pathogen interactions. So what is a host-induced gene silencing? This is a definition I gave. The genes expressed in the host plant that can interfere with the growth or development of another organism invading the plant. here we're mainly interested in the pathogens. How do we use the genes expressed in the host plants to interfere with the growth and development of the pathogen? So why should we care? The importance of this study is that first, this report was published in 2010. What they did is they have a tobacco plant expressing a small RNA molecule. That small RNA molecule is tagged in Augustine. Okay, what they did is after that is they infect that transgenic tobacco plant with a fusarium. This fusarium has Augustine expressed inside normally. So once they infect the transgenic tobacco plant, and then they find out the fusarium, the Augustine inside the fusarium is not expressed. If they put the same fusarium species on the non-transgenic tobacco plants, the Augustine was expressed normally. So their conclusion is the small RNA expressed in the tobacco plants can travel out of the tobacco, get into the fusarium, and knock down the expression of the Augustine in the fusarium. And they demonstrate the small RNA was traveling between the host and the pathogen. Once they recover the fusarium from the transgenic plants, and the Augustine is still not expressed, the suppression of the expression can last about 7 or 8 generations after the pathogen was removed from the host. And this another study is 2010 report that this similar strategy they can reduce the disease in the powder mildew in wheat and barley by expressing a pathogen effector genes, AVR10. And similar effect in 2011 that can control wheat strap rust and use a similar approach. So basically, the small RNA trafficking between the host and the pathogen can provide a new means of disease control, and which is important to us to control such as corn. However, this strategy has never been tried in corn. So this is our first attempt. So our strategy is we have several labs involved, and our lab at ASU, and a lab Dr. Deepak Patinak at the USDA is ARS in New Orleans, and Dr. Bert Brom in University of Arkansas, and also Dr. Carl Wang at Iowa State University. We're all 4 labs working on this project because each of our lab has their own expertise. So we combine those expertise here. So in our lab and in New Orleans, we have been studying the genes, the gene pathways for aflatoxin biosynthesis was first described in New Orleans. So they have knows what are the genes involved in the toxins. So our first focus is to try to identify the genes involved in the infection of a host plant. What our thinking is, if we stop the fungus from infecting the host, at least we get something done. We don't have much toxins. And that's for the first year. And the second year, we also start to look into, once the pathogen get into the host, we need to make sure they don't produce toxins. So we have also been looking into genes that are involved in toxin pathway to get silenced. And in Dr. Bloom's lab, he also has been looking for genes that are involving regulating both aflatoxin and fumonisin. So once you knock out one gene, you can knock out not just aflatoxin, maybe fumonisin as well. So once we identify those genes that are for silencing, the next step we construct the transgene vectors. And then what we did, we have tools with Dr. Bloom's lab has been focused trying to test transgenes in the fungus. And we have been collaborating with our expressed genes in corn, and then test the transgenic corn plants for the changes in aflatoxin resistance to see whether they become more resistant to F-levels infection and have a reduced aflatoxin production. So these are the outline of our strategies, and in the next few slides I will slowly give you an update on each of the areas. So, as some of you pointed out, F-levels has thousands of genes expressed. Which gene do we choose? And what I said, the first we try to stop the fungus from infecting the host. So this is our number 1 focus. And this is just one of the genes showing the cultural filtrate of F-levels growing on different cultural mediums. You will find out one of the major proteins here is the sodium 3 kilododin. And originally, we first noticed this protein is not from the culture fish, it's from this study. These are the resistant, susceptible, the cone lines. Once you infect with the F levels for 7 days and the extract proteins, and you find out most of the cone protein has been broken down into smaller now disappeared. But one of these proteins here is very dominant in the infected lines here. And we sequence this protein, we recover them and send out to peptide sequencing. And we did a sequence homology and it's identified as an alkaline protease. And I'm not sure whether Dr. Brown has found this in your proteomics. It's one of the dominant proteins in the infected concomitant. And so we clone these genes, and it has 4 exons separated by 3 introns. And we think this is one of the good types. At least it has high-level expressions in the medium and also in infected cocos. The second gene we choose is based on overlay study in 1997 by a group, I'm not sure, I think it's North Carolina. And what they found is that pectinase, we initially as P2C, is important for the infection in cotton by F levels. What they did is they have a mutant that does not express this pectinase. This is the amount of the infections on cotton locules. With that pectinase gene is transformed into it, you have an increase in infection. And this PGYF fluorescence that represents the amount of growth inside the cotton also shows when you have this pectinase, you have an increased growth of the capacity in the cotton. So we choose these 2 candidate genes. So the next step is how do we make a construct to express this gene, and the gene has to be in the double strand. This is the tricky part. And this is a complex stage. The key is try to make them double strand. We have to loop the same gene in reverse orientation. So once they transcribe, they form a hairpin structure, and then become a double strand. So all these stages involving like this is a part of the alkaline protease. We put like a 3' end here in the forward orientation, and then we have an alkaline protease gene in the reverse orientation, and separated by a intron. The intron present in there try to stabilize this construct in the construction stage. But once you put it into the plants have the mechanism to splice the intron out. So once they splice, they become together and then form a double strand. So this is the construct we designed using gateway system to make the construct. It's complicated, but once you know how to do it, the technician in New Orleans spent about less than 2 months to make one construct. It's very efficient to get this ready. This is final assembly construct about 11 kilobases. Then this is the 2 arms separated by the intron. And once it get into plants, they become a double-strand arm. Once it becomes a double-strand arm, they can initiate a sequence-specific degradation process that can produce small RNA. That's the goal. So once we construct this DNA construct, we cannot see it. So we have to somehow test whether we got the right construct or not before we send out to our stadium for transformation. The initial confirmation we did was through the restriction digestion to show different fragments of size. Each of them has a different size. We have an expected size, and we look at the geodesy, the fragment has the size we are looking for. And here we show all 3 constructs has a correct size. And so initial confirmation is good. The next thing we did, we sent out for sequencing. For sequence the construct to make sure they are correct. We did a similar strategies to also construct the pectinase gene into this RNAi Higgs vector. So I'm just kind of summarize that we selected 2 genes that are targeting on restricting the fungal growth, and we construct those vectors, and we did the restriction digestion confirm those are correct. And 3 of those positive clones will also have sequence confirmations, so they are correct clones. So once we have the clone, we have to get it transformed into maize before we can do anything. So the transformation, that's where our stadium comes here. So, and they are very good in transforming cones into genes into cone. Here, I'm just kind of summarize a lot of the events that we involve in this 2 construct. We made the side construct in this complex in March last year, and for the second contract in April. And we sent both contracts to our state university in July 1st, and they introduced into higher 2 seats and also into inverse seats. And this, we have to pay for the charges, and this was able to talk to them to do it for free for me. The condition is, if I do it for free, I won't be able to produce seeds for you. So they only give us a seedlings. So I have to babysit them once they are seedlings. We got the seedlings shipped in November 19th. It didn't ship to me. The problem with that is I don't have the greenhouse to handle the transgenic plants. This is the limitation at our university. It requires certain specifications to make sure the pollen does not escape. And due to this collaboration, somehow it's false, but it works out very good for me. Both happens to have the good greenhouse, and he was good in pollination. So we sent the material to his lab, went there for a visit, and they transferred the plants into pots in November 20th. And I got some of the leaf tissue for testing in early January this year, and he started pollinating those plants in late January. So I hope that he finished. I got all his lab busy for me. And these 2 genes, they are in our state university. They are in the greenhouse now. They are trying to produce seeds. And this is one of the things with cone transformation is a slow and also expanded stage. So one of the reasons we get the seeds to Arkansas is that we try to team with him, and Dr. Bloom is trying to see whether they can do transformation in-house. With these materials, he get the first train testing, try to see the pollination is okay or not. I think we will pretty good. This is the plants about a week after we potted, and this is the photo I took in Arkansas. And it's very tiny at the beginning, but they are big now. We did 5 independent transformation events from our, and this we got 6. And this is how many plants for each of the events they sent to Arkansas. And sometimes we only have 1 plant, sometimes have 6 or 8 plants. And this has 6 events range from 1 plant to 10 plants. In January, when we talked with Bud and he shipped all the leaf tissues for us to testing, this is the number of leaf tissues we got from him. It's very tiny. We only get about 1 gram of leaf tissues. The reason for that is the plant's so tiny, we cannot really cure the plant to get the leaves. Once we got the leaf plants, and this is really updated. Some of it I just came in yesterday. So what we did with 1 leaf tissue, we first have addressed, do we have those plants contain the target genes? So we have to do a PCR confirmation. And these are the positive control for alkaline protease. We use fungal DNA as a positive control. We use cone DNA as a negative control. And this is the band we're looking for. And we have pretty sure all of them are positive except this one. And I asked my technician to repeat this study, and it was due to not enough templates, and it's positive as well. And same as here, this is for P2C genes. We have less events in total number, so all these are showed positive, and these are the negative control, these positive controls. So we have demonstrated most of the plants we receive have the trans genes inside the leaf tissue. The second question was, are those genes are expressed? I know we cannot test the protein yet. At least we can ask, are those genes expressed? The good news is they are expressed. And this is the alkaline protein. This is the semi-quantitative. It's not as quantitative, but it's semi-quantitative using RT-PCR method. This is the 18S as a control. We use the same amount of template cDNA for the PCR. And these 2 lines have significantly high levels of alkaline protease gene expressions and than some of the other lines. Similar situation, we have high expression of pectinase in some of the lines and low in some other lines. So this is very encouraging. This is where we are now. The next step, as we are talking, I talked with Dr. Bloom last week. His student is packing up some leaves and shipped to us about maybe this week. So we can do some more testing, preferably with some antifungal assays on the leaf tissue to see we have increased fungal activities from the transgenic leaf tissues. So I'm going to summarize up here. Both contrasts have been transformed into cone, and the transgenic plants has been shipped to Arkansas, and they have already been pollinated, and the tissues has been preliminary tested. The next few slides, I'm going to talk about some of the things that has been done in Dr. Bode's lab, and I'm trying to do my best to answer. Okay, they have mainly focused on creating transgenic lines and do testing to in-house cone transformations. So in the next few slides, I will just say what are the efforts they try to do gene discoveries. They have screened over 20, identified over 20 fungal gene candidates that have been tested. One of them is here is a kinase. Once they inhibit it, this without inhibitor, this is how much aflatoxin was produced. When you soak the cones with the inhibitors, you see a reduced kinase inhibitor, you see a reduction in the toxin production. So if you inoculate it with the inhibitor, you can see also significant reductions. The other gene is alpha amylase, and they found out when you inhibit the alpha amylase, there's significant reduction in few monocytes. I want to say a little bit more. This alpha amylase, in the earlier study by Dr. Charlie Watershop's group, they have identified when you inhibit this alpha amylase, you also get a suppressed aflatoxin production. So by inhibiting alpha amylase, we may be able to inhibit both aflatoxin and free monocytes. So this is a good target to work on. They silenced 2 candidate genes, and the lower aflatoxins, these are the ones. And 11 additional candidate genes are currently being evaluated in fungus. And these are the transgenic plants that came from ours, and they have been doing the pollinations. And one of the key things I did not dare try is growing the plants in the greenhouse and try to self-pollinate them. The tricky part is to get the pollen to synchronize. And they were so skilled, they got them to synchronize. And when the plants need the pollen, they have the pollen ready. This is the important part. For transgenic plants, if you miss that part, you won't get any seeds. And they also have been doing more gene discovery focus on gene profile analysis as well. And all his students has been doing the pollination for the lines I sent to them. And this is how big they are. See, in November, they were tiny, they're this big. And I believe we got good pollinations from these students. So hopefully we'll get a lot more seeds in March or April. And also, their lab has also been doing in-house cone transformations. I say the cost is an issue. We try to stretch your funding support as far as we can. If we send to our study group, for each independent event, it's $1,000. So for each gene, I'm spending $6,000 on. That's how expensive they are. So we try to do it in-house to cut down the cost in the future. But this is also the highly technical things that take a while to get used to. So that's where we slowly try to switch to these directions. And these are the common callus tissues where you introduce the genes through gene guns and they do tissue culture to regenerate the plants. So from here, I just kind of acknowledge the people who really did the work. First is Ms. Wei. This is the supporting scientist at USD in New Orleans. She's very good at making constructs. All the complicated designs, I made it. I gave her the design. She knows what to do. She make that into the plasmid construct in 1 month to 2 months. So she's very capable. And this is Dr. Frame. It's from our state university. She put our construct in the fast track. We were able to get the construct transformed from July and get the plants back in November. So this special thing for her and our technicians at LSU and Dr. I'm not sure, Rami something. It's from Dr. Boden's lab. So he spent a lot of time babysitting our plant, and we keep his lab busy the whole time. So really, this is a true team effort to make this project happen. So I'm really excited about these things, and I want to special thanks to this funding support from AMCOE. Without this thing, nothing would have happened. And also, I would like to make special thanks for the USDAIs for the cooperative agreement. With that agreement, I was able to do more things with their funding support. Thanks again, and I'll be answering any questions you have.
Questions? On the idea that two is better than one, can you cross that P2C and